Hello, my name is Didier Stainier. I'm a director at the Max Planck Institute for Heart and Lung Research in Bad Nauheim, and my lab works on the formation and function of a number of vertebrate organs, including the cardiovascular system. Today, in this first part, we're going to be talking about vertebrate development, and specifically about the use of a model system, a zebrafish, a tropical fish, in trying to understand how vertebrate organs, including the heart, form and function. And so after a few introductory slides about the general points and general questions about development, we'll go into the various models that have been used to study development and then look to see how and why the zebrafish is helping us understand the fundamental questions about vertebrate biology, including developmental questions. So the question, simply put, is shown here. So essentially, imagine a fully grown organism as shown by Elvis Presley, who actually was stationed in Bad Nauheim. You can see that we have an organism of about 180 pounds, which consists of thousands of different cell types. And 22 years before, essentially, this organism consisted of a fertilized embryo, a fertilized egg, which weighed about 1 nanogram and, of course, was consisting of only a single cell type. And so the question is really, how does a fertilized egg become, over a period of gestation of 9 months and then a maturation period of many years, a fully grown organism such as the one shown on the right? And just to put and identify some of the questions involved in vertebrate development or in developmental processes in general, we can see a number of questions here, including cell differentiation, cell proliferation, cell death, cell motility, how the various cell types come together to form the various organs, tissues, and then finally how the organs function and maintain during homeostasis. And essentially, in the human genome, we have about 21,000 genes, and these genes are expressed in different combinations, different times, different places. And there are several questions, including which of these 21,000 genes are required for tissue and organ development, and what are their functions, and also how these genes are essentially expressed in different cells, groups of cells. And so how do we essentially address or answer these questions? And so the field has been using a number of model systems. Some of them are shown here, including the fly, the sea urchin, the chick, you can see the xenopus right here, the worm C. elegans, and then the orario, which is right here, the fish that would be the focus of this talk. And of course, let's not forget the mouse. And so essentially, these model systems have been used for many years, decades, in fact centuries, some of them, and there have been mainly two approaches to study development, an embryological approach and a genetic approach. Let's just say a few words about the embryological approach, which was, again, the main approach used for many years, starting in the late 18th century. And so a classical experiment done here in Germany by Mangold and Spemann in the early 20s consisted of essentially taking part of a dorsal tissue, as you can see here, and putting it on the ventral part of an early stage embryo. And that once the embryo was raised, essentially you can see here at the bottom that there was a second axis formed on the ventral side of this embryo. And this led to the concept of embryonic organizers, whereby tissues or groups of cells were secreting growth factors that were inducing adjacent tissues to form specific structures. And much work has been done since then to identify not only the genes, but also the mechanisms involved in this tissue induction, not only from this dorsal organizer, the so-called Spemann organizer, but also other organizers. The other major approach that's been used essentially to study development is a genetic approach. And so forward genetics and reverse genetics have been used traditionally. And just to first define these terms as they'll be used throughout these talks, forward genetics is a means by which essentially mutations are induced randomly throughout the genome. These mutations are homozygous, and then the organisms are screened for interesting phenotypes. In contrast, reverse genetics consists of specifically focusing on a single gene or several genes. These genes are specifically mutated, and then the homozygous mutant organisms are analyzed for interesting phenotypes. So let's focus a little more on forward genetics, and to use a phrase used by many geneticists, including the late Ira Herskowitz, the awesome power of forward genetics has really led us to many insights about key biological processes, including, for example, from the yeast, shown on your left. We have gained tremendous insight into the control of the cell cycle. Forward genetics in C. elegans has given us insights into mechanisms of apoptosis or cell death, as well as the discovery of microRNAs, and of course the logic of embryonic patterning and developmental processes has come from forward genetics in the fly. And what about vertebrate organisms? Certainly vertebrates have organs that are not seen in these invertebrate model organisms, and also many genes that are specific to vertebrate species. And so the idea then is, can we identify a vertebrate organism in which to do such forward genetic screens? And certainly there's been the mouse, many screens done in the mouse, again, since many years. And these screens have certainly led to many insights, including, for example, about the signaling function of these specialized structures called cilia. Now, doing forward genetic screens in the mouse is a fairly complex and expensive process for a number of reasons. And of course, the litters, the number of pups that one can get from single pair matings of mice is fairly limited. And so in the 80s, this tropical fish, Danio, which originates from the Ganges in India, became an intriguing model system that was possibly amenable to forward genetic screens. And the main reasons that Danio seemed to be a good model is shown here. And so from single pair matings, you can get large number of embryos, which of course for genetic screen is key. And also in terms of development, you can see that fertilization is external, which means essentially then that one can visualize and manipulate these embryos from the onset of development. And this of course is very important in terms of trying to observe development as it takes place. And so a typical fish room is shown here. You have of course many tanks, and each housing a different genetic strain. You can also see right here the breeding boxes that are used, and you can separate the males and females from their eggs. And essentially this brings us to not only are these embryos fertilized externally, but the development of these embryos is also very rapid. So within 24 hours, the heart has started beating, and by 48 hours, most of the organ systems are in fact functional. So not only that, but these embryos are also transparent or translucent, as well as the larvae. And so these and other attributes of this system really allow us to do not only forward genetics, but also use embryological approaches and really try and get to a cell biological understanding at single cell resolution of developmental processes. I should also mention that the zebrafish community is become a very large community, more than 1,500 labs, and so far more than 30,000 papers, and as I said, started around in the early 80s. So I'll now show you a movie of about the first 24 hours of zebrafish development, and before that, let me just show you a few key developmental stages. So on the panel A, you can see that in fact we have the one cell stage embryo, which consists of a cell sitting on top of a yolk mass. The yolk, of course, is being used for nutrition prior to the start of feeding. You then have a series of rapid cleavages to give you the blastula stage, and then gastrulation starts, and this is when the germ layers, the ectoderm, mesoderm, and endoderm, are being formed. Within essentially 12 hours of development, your gastrulation completes, and then you have the formation of the somites, shown right here. By 24 hours, as I showed you, the heart starts beating, and then by 48 hours, most of the organ systems are functional. So this is the movie again, starting from the one cell stage. As I said, a series of rapid cleavages forms the blastula, and then the gastrulation starts. during a process called epiboly, by which the cells surround the yolk mass. You have the somatogenesis, and then at 24 hours, the heart starts beating. So just to focus on a few of the organ systems clearly visible in the fish, you can see on the top panel right here, the heart, which is very prominent and will be the focus of this first and second part. You also see the vascular system, not only the main vessels of the trunk, but also the smaller vessels that are formed through angiogenesis. And then finally, and this is now using a transgenic line, and we're looking at the liver, the pancreas, and again I'll show you a confocal stack rotated to give you a sense of the resolution one can achieve using the simple imaging techniques and this model system. So again, we have the liver right here, and the pancreas right here, and then this is the hepatopancreatic duct connecting the two. And again, I'll just rotate this movie. Behind these is the swim bladder, which is a buoyancy organ for the fish. And again, you can get a sense of the single cell resolution that one can achieve for these vertebrate organs. There are two main advantages to the zebrafish model as listed here. The first one is the live imaging, as I've shown you before. And the second one is the ability to do large-scale screens, taking advantage of the fact that one can get a large number of embryos from single-pair matings. And this is both now for genetic screens as well as small molecule screens or chemical screens. So let's first look at the live imaging by focusing on the heart and take a historical perspective. And so this is the way the heart was looked at in the early 90s, which was when the initial large-scale screens were done. There were two large-scale screens done, one here in Germany by Janine Nusslein-Vollhardt's lab in Tübingen, and the other one by a former student of hers, Wolfgang Drewer, who then was leading a lab at MGH in Boston. And essentially, simple white light microscopy was used to be examining and screening these embryos, as shown here. And we'll come back to this organ system later on. So in 2004, people then started taking advantage of fluorescent transgenic proteins, including GFP. For example, here, GFP is labeling the myocardium, or the muscle cells of the heart. And it took a few years later, and a special microscopy technique was revived called selective plane annihilation microscopy, or SPIM. And you can see here again that we're looking at the heart, again looking at the cardiomyocytes in the muscle cells of the heart. And you can see the blood flowing essentially from the yolk mass, around the yolk mass, into the atrium and then into the ventricle. The atrium being the collecting chamber of the heart and the ventricle being the pumping chamber of the heart. Here we're looking at just a single cell layer, but of course the heart consists of multiple cell layers as shown here. So now we're labeling in dark red the myocardium, in bright red the blood, and in green the endocardium, which is the endothelial layer inside the heart. And again, we're looking at single plane images using SPIM imaging, but of course the heart is a three-dimensional organ. And so now taking advantage of spinning disk microscopy, followed by image reconstruction, you can see, imagine you're standing inside the ventricle looking out, and what you're seeing are these muscular ridges here. Essentially, this is the best seen here, and these are called trabeculae. And let's just come back to the previous slide. And again, these are these ridges that we're seeing here in cross sections, and these are the trabeculae. And we'll be looking in the second part of this series, we'll be looking at how these trabeculae, these cardiac trabeculae, are forming. So essentially, we also, of course, want to look at these organs in three-dimensional and with time, but also at a single cell level. And so, for example, we and others have generated transgenic lines where one can look at the actin filaments here on the right, or sorry, on your left, or the microtubules here on your right. And we'll be coming back to this actin filament transgenic line in the second part of this series. So essentially, as I said, imaging has been a key part, a key tool for zebrafish heart research, not only in terms of visualizing the events that are occurring, but also in terms of really leading to a hypothesis that then can be tested either genetically or embryologically. As I mentioned, forward genetics was the main means by which people were identifying mutants and interesting genes that essentially regulated the process of heart development and function. The morpholinos, which are modified and stable oligos that are used as antisense reagents, were essentially came about in 2000. We had a very efficient transgenesis using the TOL2 transposon around the same time, chemical biology, and we won't say very much about that around 2003, and then reverse genetics using zinc finger nucleases, TALENs, CRISPRs, essentially just a few years old now. Now, in the third part of this series, we'll be looking to see how, in fact, when people were using morpholinos, these antisense reagents, to look at gene function, they identified a number of phenotypes, and some of these genes that were analyzed using morpholinos were revisited using these reverse genetic techniques, mainly the TALENs and CRISPRs, and very often the phenotypes resulting from these two different approaches appear to differ, with the mutants phenotype being much milder, and so we'll be trying to understand how some of these differences may occur. So, as I mentioned, besides the imaging, the other major advantage of the zebrafish model is the ability to do large-scale screens, be they genetic or chemical screens, and I'll just focus here on the genetic screens. And again, just focus on the heart as the model system. So I've shown you this movie of the wild-type heart, and on the right, you can see a mutant heart. This mutant is called cardiomyocytes, and what you see is that the blood essentially moving back and forth between the two chambers, the atrium and the ventricle. And it turns out that this cardiomyocyte mutation affects an actin gene that's expressed in the myocardium. And this leads to aberrant contractility of the myocardium, which in turn then leads to defects in the maturation of the heart, specifically in this case in the formation of the valves that prevent retrograde blood flow, and I'll have more to say about that in a few minutes. But the point here is that the concept of this tight interaction and crosstalk between formation and function is, of course, present in many organ systems, but specifically prominent in the development and formation and function of the heart. So one can, of course, get more sophisticated in terms of analyzing the various phenotypes and also identifying mutants with interesting cardiac function defects. And here, for example, I'm showing you GCAMP, a GCAMP line which is used to look at the calcium flux. And GCAMP is a modified GFP protein. And again, you can see in this case right here is the ventricle, and you can see these muscular protrusions, which are the trabeculae. So, for example, we identified in one of our screens a mutant that we call dococ, and you can see again the ventricle beating away, and in the mutant you can see that this ventricle is fibrillating. And one can look at this more closely using this calcium line that I introduced earlier. And essentially you can see that there's a partial block along essentially the left side of the heart. The right side of the heart appears to propagate the calcium wave normally. You can see this much better if you look at, or much more easily, if you look at an isochronal map. It's again a partial block along one side of the heart, and this mutation affects the connexin protein, and when a mutation in the mouse ortholog was made, a similar phenotype was seen. So again, suggesting that in fact the mutations in the human gene may also lead to the same kind of phenotype that is seen in this zebrafish mutant. So with that, I want to come to the last point of this lecture, which has to do with the endothelial layer. And the endothelial layer is a vertebrate invention, and here we're looking at the endothelium in green, and we're looking throughout the embryo. We're looking at the blood cells moving through the vasculature. These blood cells are labeled in red, and you can see right under the eyes, you can see essentially the heart pumping away. And essentially, as I mentioned, the endothelial layer is a vertebrate invention and was key in terms of the evolution of the cardiac pump. And so if you start with, on the left, what is a simple chordate, the heart of a simple chordate like a tunicate, and this chordate lacks endothelium, and essentially this heart can pump blood in both directions. It's more like a peristaltic pump. And as you move to look at, for example, the fish heart right here, you can recognize a number of new features, including the subdivision into chambers, the atrium, the collecting chamber, and the ventricle, the pumping chamber, as I mentioned before. There's also specializations of cells at the boundary, the AV boundary, the atrioventricular boundary, both in terms of the myocardium, the muscle cells, as well as the endocardium, the endothelial cells. And then you may have seen from the movies that there's a coordination contraction in the sense that the atrium contracts first, then there's a slight pause, and then the ventricle contracts. And of course, moving then from fish to amniotes, of course, there was a subdivision of both the atrium and the ventricle, leading to the four-chambered heart. So, as I mentioned, the endothelium, which is known as the endocardium in the heart, is a vertebrate invention. And it allowed a number of features, again, listed here. And we'll go through three of them in turn. So it allowed a more efficient pump by allowing the complete occlusion of lumen. It directly participated in the formation of valves between the chambers to prevent the retrograde blood flow. And it allowed the increase in muscle mass by providing the signals for the formation of trabeculae. And this is prior to the formation of the coronary vessels, which feed the muscle wall. And these trabeculae are also, of course, key in terms of the formation of specialized conduction system, but we want to say more about this today. So let's start with the fact of this more efficient pump. There was probably the initial selective advantage for this appearance and selection of the endocardium in the heart. So imagine that, again, starting with a simple heart, like the tunicate heart, which consists of a single cell type, which are the muscle cells. As these muscle cells contract, they lead to the formation of a finite lumen. Now, if you bring in a second cell type, which are the endocardial cells, and assume that these cells can be distorted or deformed, as the outer layer contracts, then this inner layer will essentially completely occlude the lumen, and presumably then allow the complete emptying of this chamber. And in fact, if you look at the zebrafish heart, and again, this is SPIM imaging by Jan Huisken, you can see that indeed in the green you can see the endocardium, and as the red cells contract, and here we're looking at the atrium right here, as the red cells contract, the green cells come together, completely occluding the lumen. And you can see this better by just focusing on the endocardium and see how these cells, and again, of course, we're looking at a single plane, but these cells are coming together and from the three-dimensional organ, and you can then lead to the complete occlusion chamber and complete emptying of this chamber. This is not a new concept in the sense that the cardiac jelly, which essentially is a specialized extracellular matrix that lies between the endocardium and the myocardium, had been proposed to play this role in terms of if you assume there's a finite and stable volume of cardiac jelly, then of course as the outer layer, the myocardial layer, contracts, then this will also help lead to the complete occlusion of the lumen. So moving on then to the second point about the evolution of the cardiac pump and the fact that the endothelium was key in terms of the evolution of this organ, I will show you how, in fact, it is directly involved in the formation of the valves between the chambers. So let's first look at the retrograde blood flow as it appears at early stages. So as I mentioned, the zebrafish heart starts beating at 24 hours post-fertilization. And you can see at 55 hours, you can see again, this is the atrium right here, and the ventricle right there, and the atrioventricular canal right here. And you can see that as the ventricle contracts, a few blood cells are moving back towards the atrium. Now, if you look at 70 hours, you can see, in fact, that there's no retrograde blood flow, and you can also notice that there's now a thickening of the atrioventricular canal, essentially that helps to prevent this retrograde blood flow. And the shape of these cells is diagrammed right here. So essentially, again, looking at this more closely, one can see that, in fact, there's the AV canal, as you can see right here, there's these cells, the endocardial cells, have assumed a different shape now and are forming what looks to be a S-shaped structure, possibly through invagination of these cells or partial epithelial to mesenchymal transition, as we've gained evidence for, and essentially then these structures can then get remodeled into these valve leaflets that you can see now very clearly at 7 days, again, between the atrium and the ventricle. And you can see that these valve leaflets are very productive and very efficient at preventing retrograde blood flow. So essentially, then, one of the key questions that we and others are trying to address is essentially trying to get a single-cell resolution understanding. And again, we're looking here, this is the atrium and the ventricle, and the atrioventricular canal. And you can see in green, again, we're labeling the endocardial cells, and you can see these are single endocardial cells right here. And you can see how essentially these cells, as they acquire a different shape, and as various genes and mechanisms are essentially affecting their behavior, these cells are now forming these specialized structures called valves that prevent retrograde blood flow. And so this is really the goal of this, investigation now, to understand, not only identify the genes responsible for the formation of this structure, but really understand how these genes drive the behavior of each cell as it forms this evolving structure. And very similarly, in part 2, what we'll be looking at is this process of cardiac trabeculation, as I mentioned. And again, as you can see in this cartoon, trabeculation is an interesting process. As you can see, trabeculae are mainly formed here in the outer curvature of the ventricle, but not in the inner curvature, right here, and also not in the atrium. And so we'll look in not only in terms of the cell behavior of these cardiomyocytes as they form this cardiac trabeculae, but we'll be looking at some of the genes, and also, again, coming back to a process called epithelial-mesenchymal transition. And then in the third part, and as I mentioned, the original way that genes were analyzed in zebrafish was through forward genetics, and then people started using, about 10 years later, they started using these antisense reagents and analyzed the function of a large number of genes. And when the reverse genetics techniques became available just a few years ago, some of these genes that had been analyzed, whose function had been analyzed using morpholinos, were revisited using TALENs and CRISPRs. And essentially the phenotypes resulting from these mutations were often milder than the phenotypes that had been observed using these antisense reagents. And so we're trying to understand what may be the basis for this discrepancy between antisense-induced phenotypes and mutation-induced phenotypes. And this will bring us to essentially look at the question of mutant versus antisense phenotypes. And with that, I'd like to thank the many past and present members of the lab, as well as the funding agencies, both when the lab was at UCSF and more recently at the Max Planck Institute in Bad Nauheim. Thank you.